Hello, my name is Jack Weatheritt, and I'd like to present some work that I have been doing with my co-workers at ICSICO and Imperial College London. The title of my talk is Transfer Learning for Brain Segmentation, Pre-task Selection and Data Limitations. So we'll be looking at deep learning approaches for anatomical brain segmentation, and specifically how can we utilize this concept of transfer learning and how much data do we need in such a pipeline. So I'll start by introducing motivating the problem from a clinical and computational point of view. We'll look at the data sets that were used in this talk, and then I'll introduce the workflow, the deep learning and transfer learning pipelines, and then we'll look at some results and finally some conclusions. So the clinical problem of interest for us is accurate delineation of the caudate, which you can see here on the right-hand side in this MRI scan. It's of particular relevance in Huntington's disease, where the volume change over time is a biomarker for the progression of the disease, and also to the volume change can highlight any efficacy of drugs taken throughout a clinical trial. Therefore, accurate segmentation of such a region is of clinical relevance. The current gold standard is to use an expert to manually segment the region. However, this is time consuming, and the scale of clinical trials can make this too costly. Instead, what we're exploring is using deep learning approaches to segment the caudate that look like something like this. We have a neural network here, schematic on the right-hand side, which typically for such a problem consists of several 100,000 parameters that need to be trained. This training typically requires gold standard training data for which you may not necessarily have for a specific task. So then the problem becomes, how can we use the data intelligently that we have, and how much data do we need for a specific problem in order to get accurate predictions? The general motivation then is to minimize the number of manual labels required in order to train a network, and in doing so we're attempting to utilize data that we already have, and the output is a pipeline that can accurately segment the caudate. And this is kind of a simulation of a new trial, the beginning of a new trial for which we might not have ground truth caudate segmentations for a specific population or a specific scanner protocol. So I'll now introduce the workflow and the data that we are feeding into our pipelines. So we have 5 tasks on this screen here, and these are the data sets that we've used throughout. The target problem is the caudate segmentation for an HD population, so it's a HD trial here, and we have gold standard segmentations outlined in blue for subjects collected in MNI space. For the same set of subjects, we have lateral ventricle labels collected in the same space, so that forms a related task but a different anatomical structure. Again, for the same subjects enrolled in the trial, but now in native space, we have whole brain masks manually segmented. Then on the left-hand side here, we have a different disease population. So these are Alzheimer's patients in the ADNI cohort. And using this data, we've applied the MALPM pipeline to get the automated labels of the caudate. So this is the same ROI as our target distribution, but a different disease population, and also the labels are automatically gathered, not manually labeled. Finally, we use the OASIS data with the Neuromorphometrics Atlas to provide a validation set. Now, we're not specifically interested in the quality of predictions on this data, but some interesting points can be raised by testing our models on a completely unseen data set. So this is our workflow here. If we consider the middle row, say, the basic premise is to take one of these pre-tasks. So in this case, this is the HD trial, ventricle labels. And we begin by training the entire network on the lateral ventricles, the manually segmented lateral ventricles. We then freeze the left-hand side of the model, so these weights can no longer change. We then apply the same model in a new training process where we only train the last layers of the network, and we apply it to our target data, a subset of our target data set, and this is the manual segmentations of the chordate. Once we've tested the model in this distribution, we can then validate the model on the OASIS dataset. Now we have 3 different pre-training tasks. We have the lateral ventricles, which I've just gone through, but we can repeat the process with the whole brain problem and also the automated labels gathered on the ADNI dataset. The final model to compare against is a model that did not receive any pre-training, so we can ascertain the importance of pre-training by not doing any, and then training an entire model on a subset of the target problem, and then validating as well on OASIS. So the pre-processing that we use, we first begin by affinely registering to M&I space. These are the problems which require registration. We normalize our intensity to have 0 mean and unit standard deviation. The next step is to extract a bounding box around the ROI. So obviously this box varies in size depending on the structure and depending on the task. So the lateral ventricle input field is bigger than the chordate, for instance. And also the data trained in native space, so the ADNI data, the bounding box is much bigger because the head is not rigidly registered to the same space. The whole brain is different again because we feed random 64 cubed patches into the network to train. The final thing we do is to augment the data set by flipping left to right. So the model itself is a 3-dimensional unit, which means we're applying convolutions in all 3 dimensions of space. Because we don't have any fully connected layers, we can vary the input size, which is obviously crucial because we are considering different ROIs. And we have 3 skip connections, which this particular architecture results in about 200,000 parameters. For regularization, we're using batch normalization, and everything is implemented in TensorFlow 1.15, and we're using the Adam optimizer. For the fine-tuning process, this should say the fine-tuning freezes the left-hand side of the model, sorry. So during the fine-tuning, all of these parameters are fixed, and all of these parameters are freely allowed to vary when we fine-tune the model using the target problem data. So now we move on to the results. The first thing we can check is the pre-training task performance. So we're going from top to bottom. The whole brain performance has a dice of 0.97 with a very small standard deviation. Similar scores for the lateral ventricles. These are both trained on manual labels. The chordate, the automated labels on the ADNI data, the model was able to predict with a dice score of 0.91, which is a little bit below gold standard for this particular ROI. However, I believe this is down to the fact that it's learning automated labels, which themselves will have some inherent variance. Now, once we've taken these pre-trained models, what we can do is fine-tune the model on the target core data problem. And we can use a subset of that data set. So here you can see the top row are models fine-tuned using 2 brains. The middle row is fine-tuned using 16 brains. And the bottom row is fine-tuned using 306. And the ground truth is the left column. The model on the second column has received no pre-training, so it's trained purely on 2 brains, 16 brains and 306 brains from the target data set. And then we have our 3 pre-trained models with a schematic above that you can see here. The main thing to notice is that the end-to-end model, so the red and the green is ground truth, and then the red and the blue is the model prediction. You can see that the model trained end-to-end and the whole brain have performed very badly, whereas the model trained on the automated labels from ADNI has performed pretty well for a very small number of brains in the fine-tuning data set. And then, as you see, as we increase the number of data points in the fine-tuning set, all of the models are converging to a pretty similar prediction. We can accumulate this into some statistical results. So you can see here as a comparison, this is the IOU, so the Yaqub index. You can see here the red is the end-to-end model. So this received no pre-training, and for a smaller training size, it performs the worst. And as we increase the number of brains in our training set or our fine-tuning set, all of the models are asymptoting to a similar IOU score. The point to note here is that the model pre-trained with the automated labels and then just 2 or 4 brains from the fine-tuning set is actually enough to reach near state-of-the-art performance without any manual labels, without any more than 4 manual labels. This is quite surprising, actually, because this model here was trained in native space, whereas the ventricle model, so the next one along, was actually trained on precisely the same subjects, was pre-trained on the same subjects, but for a different ROI, and it performs worse than this model here. We can then look at the generalization performance. Now, actually, on the previous slide, we saw that the training on the core date was more important than training a model in the same space, even on the same subjects even. Now, we actually see the reverse. When we go to a completely unseen data set, the models with a MNI pre-task, or the model trained only in MNI space, is performing better, and the OASIS dataset is in MNI space, to say here. Whereas the models pre-trained on the native space are performing no better than random until we have at least 100 brains in our fine-tuning dataset. So that we can conclude that the MNI methods are outperforming natively trained methods. And it looks like that the larger the data set, the encoder becomes irrelevant, and the model is actually learning a completely new distribution, of weights. Okay, so I'd like to conclude here that accurate core date segmentations are possible for a specific data set with as little as 2 data points. When fine-tuning, matching the tasks is actually more important. So pre-training on a core date data set is more important than training with a specific input space. When applying fine-tuned models to an unseen data set, the original space became more important again. Thank you very much for listening to my talk. And if you have any questions, please don't hesitate to contact me. Thank you.